Welcome to Health Professional Radio. I'm your host, Neil Howard. Thank you so much for joining us for another segment. Carisma Therapeutics Incorporated recently announced that the first patient has been dosed in the Phase 1 multi-center clinical trial for CT-0508 for cancer immunotherapy, specifically in solid tumors. Well, Dr. Debora Barton, she's the Chief Medical Officer at Carisma. She's joining us on the program today to talk about this new approach in gene-based therapy development. Welcome to Health Professional Radio, Dr. Barton. Thank you for joining.
Thank you so much for having me.
Give us a little bit more about your background and talk about how that led you to your current position there at Carisma briefly.
Absolutely. So I'm a medical oncologist, and I have dedicated my life into finding new options for cancer patients. And I moved into pharma industry with the same scope, trying to go into research and trying to discover new things more than improve current things that are available for cancer patients. So I have worked with innovation in radiopharmaceutical world and then cell therapy with tumor-infiltrating lymphocytes. And Carisma came up as a very different perspective with immunotherapy and cell therapy, specifically aiming to address an unmet need of solid tumors, which is my passion. So it was very natural that I would be drawn to joining Carisma because of its platform and its goals of really addressing solid tumor patients.
Now, many of our listeners may be a bit familiar with CAR T. CAR T cell therapy has basically revolutionized how cancer is treated and revealed the power of gene-based therapies. But I do believe that it has met with some challenges with solid tumors. Talk about this therapy and relate it to the therapy that you are studying.
Thank you for that question. Yes, so CAR T cells really are groundbreaking for hematological malignancies, and they are providing fantastic results for those patients. The characteristic of those diseases is that they have this one marker that is expressed by virtually all cells. So the CAR T cell targets that one marker in the cancer cell and kills them. And the cancer cells are very homogeneous. They all look the same. And that's why CAR Ts are so successful in those diseases. The difference here is that the solid tumors are heterogeneous. So within a tumor, you have some cells with the target, some cells with a little bit of the target, and some other cells don't have the target at all. Mm-hmm. So the CAR T cells, they will attack those cells that have the target, but the tumor cells that don't have either enough of the target, or if they don't have the target at all, they will eventually outgrow. Mm-hmm. Now, the difference with the CAR macrophages is that the CAR macrophage will take that cell with the target and engulf that cell and process all the different targets that are in that cell, not only the one that is identified by the CAR macrophage. So the heterogeneity of the different targets in a tumor cell will be expressed, and the macrophage will then call for the help of the person's own T cells, and they will take care of killing all the other tumor cells that don't have the CAR target, but they have other targets that have been processed by the macrophage and presented to the T cells. So it's a two-pronged approach. I call the CAR macrophages the multitaskers. They not only kill, but they also call for help, and they coordinate an attack.
Talk a little bit about this phase 1 multi-centered clinical trial for CT0508. The first patient has been dosed. How many patients in this trial are you planning, and what type of solid tumor is specifically being looked at in this trial?
Absolutely. So we are looking to enroll 18 patients with HER2. HER2 is the target that we're looking for. So those patients need to have a HER2 overexpressing solid tumor. And when people think about HER2, they naturally think about breast cancer. But it's important to know that HER2 is overexpressed in a wide variety of tumor types, including breast, lung, gastroesophageal, colorectal, biliary, pancreatic, ovarian, and multiple others. So as long as in the biopsy it shows that that tumor overexpresses HER2, that patient potentially can be eligible for the clinical trial. So we're going to enroll 18 patients currently in 2 clinical sites, the University of Pennsylvania in Philadelphia and the University of North Carolina in Chapel Hill. And the study is open and ongoing. As you said, we successfully treated the first patient. So that's a major milestone, not only for the company, for the cell therapy field as a whole. The first time in the world a patient received an engineered macrophage to treat advanced cancer.
What's next for Carisma? You're a very unique therapeutic company. What's down the line for you?
So we have a lot of efforts going on. The phase 1 clinical trial is aiming to really prove the concept and show how the CAR macrophages work in a cancer patient. We are always striving for better and looking for improvements. So one of the very important efforts is the development of CAR monocytes. Monocytes are the precursors to macrophages. So that means that the manufacturing process for CAR monocytes will be shorter than CAR macrophages. And the manufacturing of CAR macrophages is already shorter than the regular CAR T cells. So it can be something really in a 1 to 2 day time frame. And we are also looking at allogeneic strategies off the shelf.
Well, give our listeners a website where we can learn more about Carisma Therapeutics Incorporated and about the clinical trial that we've been discussing here today.
Yeah, so you can find information at www.carismatx.com. And there you can see our pipeline and our mechanism of action. You will see that we're also developing PSMA, mesothelin, and other targets in the preclinic. And for the clinical trial information, you can go to www.clinicaltrials.gov, and in the search field, you type CT-0508, and you will find details of our first-in-human phase 1 clinical trial.
Dr. Barton, I appreciate your time here on Health Professional Radio. Thank you so much.
It has been a pleasure. Thank you for having me.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Dr. Debora Barton, Chief Medical Officer at Carisma Therapeutics Incorporated. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes. Listen in, download it, SoundCloud, and be sure and subscribe to our YouTube channel at youtube.com, Health Professional Radio.